As you well know, plants can get pretty big, and what you're looking at there is a fisheye lens photograph of a sequoia in Yosemite Park in the United States. Very, very big trees. And I have a question for you regarding trees and growth. Now, if I were to take a nail and hammer it into the side of the tree, and then go away and come back after a few years, would the nail be at the same height or will it move higher? So it's an interesting question and it helps us understand a little bit about plant growth. So will the nail be higher? Now we'll come back to that. If I look at the bark of a tree and as we say, wild human markings, someone scratches their initials in, maybe the girlfriend or the boyfriend for that summer or whatever. In a few years, you come back, is it in the same place or did it move up? Bears in the wild, obviously, they rely on marking trees for establishing territories. So if you see marks like that on a poplar or a beech, just understand that you're in a bear's territory. So make sure you pack your food up and hang it from a high branch between trees. But you think those claw markings go up or do they stay at the same height? Now, with this motorcycle, this person obviously never bothered to fix what could have been a fun old Honda. Uh, the tree grew around it. Now, it didn't pick the bike up with it as it got higher, did it? So if we go back to the initial question, the nail actually stays in the same location. It does not move up as the tree gets taller. Um, how trees grow is a lot like a Russian doll. I don't know if you've ever seen one of these. In the middle, you've got like a little doll, and as the tree grows, it forms rings around the. Let me get my pen here. I was supposed to draw on it and it moved the screen to the next one, but that's okay. This little doll in the middle is the initial tree, the sapling, and then every year it lays down more tree around it, and it just gets bigger and bigger. But the original tree is in the middle of it all, right? That's kind of cool. We're used to looking at trees and rings and things like that, and this cross section is showing us, if you cut a tree down, you see the rings. Now, what's interesting about this is if I go to let me get my pen again, you'll notice that there's bark on the exterior, right? And then we've got all this hardwood. And it's not like there's hardwood and then bark and then hardwood and then bark and hardwood and then bark cycling through. The bark always stays on the outside, right? So that also gives us a hint in terms of how plants grow. In this case, how trees grow. So if we go back to the analogy of the Russian doll, the initial sapling is in the middle here, and the tree is laying down layers around it as it grows every year, more layers. And if we cut the tree down, we're counting the rings here. So bark, on the other hand, forms on the exterior. So where's the growing layer in a tree? It's in a layer we call the cambium. So within that cambium is a specialized tissue called a meristem. You're going to hear more about that as you go through this course and through your program. The meristem is a really important part of any plant because that's where we have a specialized area of rapid cell division and elongation. So there's something for you to remember for your quizzes and midterms, the definition of a meristem. This picture, which you'll get to see under the microscopes in your labs, depicts where a meristem would be. And this is the growing tip of the plant, in this case the tip, protected around a layer. In the middle is that area of rapid cell division and elongation. That's the shoot, if you want to think of it, as the plant is growing. So there's lots of cell division happening in there. Back to the tree, in that example, the meristem, because the tree, we're talking about not just the top or the apical, we're talking about the lateral meristems, it's growing sideways. And this red ring is where we're getting cell division, and it's pushing bark out as it grows and leaving wood behind in layers. So this bark has elements of really old bark and new bark as it's sort of moving. And obviously bark helps us identify trees because depending on how the bark is shaped and how it's laid down, it sort of like tears and pulls and shrinks and stretches in different ways depending on its corkiness and stuff like that. But the meristem is just below the bark. That's why if you damage the meristem and behind the bark, you can kill a tree, right? That interferes with everything from cell division in that area to cutting off the vasculature where the sap is flowing. So it's a really important thin layer around a tree. What's also interesting about that is that when we're looking at a tree, there's actually not a lot of living material there, especially in the winter, because this wood is dead, right? That's all dead. And the bark on the outside is dead. It's just this thin layer between the two that we call the cambium that's actually alive. Um, so that giant sequoia you see is not a massive organism per se, because so much of it is just dead wood and bark. And in the case of a sequoia, the bark is incredibly thick, 2 to 3 feet thick, because it's designed to insulate it from fires. So, anyways, that's an introduction to cambium. We're going to look at that in the lab with slides. You're going to talk more about it as we go through the structural anatomy of plants. Now, with respect to the meristem, I already mentioned that there's these two processes that are happening in the meristem. We have cell division and we have cell elongation. I want to look at cell division to start with, all right? And so at the very least, we're starting with one cell and we're duplicating everything in that cell to create what we call two daughter cells. And that process is called mitosis. Some of you may have learned a little bit about this in high school, some might not. Um, that is the process of copying the DNA in the cell. Now, a lot of the other structures have to be copied too, but we'll concern ourselves mostly with mitosis. Um, the second part of growth entails elongation. So over here we have cell division, right? So where we go from one cell to two cells. And after they divide, typically the cells elongate or they stretch. And that both those processes are involved in growth. And trees and plants in general will elongate through a process called osmosis. So using water pressure to sort of hydraulically press the cells out into a larger shape. So we'll touch on that next. Let me just change slides. So mitosis, taking one cell, copying all of its DNA, and then producing two daughter cells. That whole process of cell division. Um, DNA is like a library and it's got all the books. And in order to be able to rebuild a new cell, you need to have all the books. Otherwise, you're missing important information. So it's really essential that the DNA is copied accurately in order to have two daughter cells that are viable. Now, out of interest, I put over here on the side that in order to make DNA, you need a lot of nitrogen and phosphorus. And that is one of the reasons why we need so much nitrogen and phosphorus in fertilizer, because the DNA is made up of a lot of nitrogen and phosphorus. And if you don't have enough of that fertilizer, the cells can't properly divide and remain viable. So really important that plants have proper nutrition. I mean, we knew that, but we're just giving a glimpse again into why that might be the case. Uh, so yeah, like I've said, DNA is sometimes referred to as the blueprint for life. I'd like to think of it as a library. You know, it's a whole bunch of books, and each book is specific instructions on how to build and how processes function for a living plant cell. So as plants grow, they need more DNA, right? So if a plant wants to become bigger, it needs more cells, it needs more DNA. And so this process of mitosis is how that happens. Um, and one of the first steps in a cell duplicating its DNA is it has to, first of all, copy it, but then to keep everything organized, the DNA has to be sort of coiled up into what we call a chromosome. Um, Normally, if you are looking at DNA in a cell that's just going about its regular life, functioning, normal business, the DNA looks like just sort of a mush of a stringy solution. An analogy would be like a bowl of noodles, just all over each other. And when you're trying to pull the noodles out, they get caught and tangled, and it becomes a nightmare. So what happens is, as the bowl duplicates all the noodles and they're now copied, then what it does is it sort of coils up each noodle until it's a chromosome. And another really good analogy I have for understanding what a chromosome is if you take an elastic band and you start twisting it, right? You twist that elastic band and you keep twisting and twisting and twisting. And you can try this at home. You'll see exactly what happens. This is how chromosomes are made. The elastic band will start to spiral on itself, and then it will collapse, and then it will spiral again. And you get higher and higher levels of spiraling until you have a really dense coiled piece of rubber band. And that's essentially what a chromosome is, right? It's DNA, like think of this thin, long strand. And by the way, a typical plant chromosome, if you stretch the DNA molecule out, is about a meter long, which is astonishing when you think that that thin molecule fits inside a nucleus. And plants have anywhere from 12 to 40 chromosomes in there. So that's a lot of DNA, which is again why I said you need a lot of nitrogen and phosphorus for the plant to be able to recreate all that DNA. Anyway, so the DNA is spun up and condensed into a chromosome. And chromosomes have sort of common structural elements. Now, the arms, or in this case, chromatids, can have different lengths. So chromosomes don't all look the same. And sometimes in biology, we rely on that because when we look at chromosomes under a microscope, it can help us identify which chromosomes are what just by looking at the size. But for our purposes in this course, that's not something that we're doing. But we'll have slides so you can see chromosomes. But the chromatid is an arm. The center is what's called the centromere. And then, of course, the arms are split into short arms and long arms. The centromere, interestingly, is kind of an anchor point so that once the chromosomes are formed and they're ready to be assigned to their new daughter cell, there's little tubes that anchor onto the centromere and then start to retract the chromosomes to each end respectively. So the plant has this really complicated process that it has to go through to make sure that A, the DNA is copied accurately, that B, the chromosomes are formed and not tangled up, and then B, those chromosomes have to be organized and then arranged in such a way that when they get pulled to the different daughter cells, or the two subsequent cells that are being formed, that all the correct chromosomes are in each cell. Because if you end up with a cell that doesn't have all the correct chromosomes, it may have a bunch of DNA, but it's missing a bunch of books in its library. And it may not know how to build half the proteins that it needs in order to live, and then that cell will die. So it's really important that the chromosomes are organized. And so in mitosis, on your own, of course, you can watch this video that might help you comprehend how this stuff flows. So rather than me trying to recreate it, I would highly recommend you click on just the PowerPoint slides of this and then click on that link. But mitosis is broken up into 6 different stages. There's an acronym down here that will help you remember for your midterm, IPMAT, and it starts with what's called interphase, which is where the DNA is being copied. So it's still like a bowl of spaghetti noodles. Prophase, where the DNA starts to wind, or we call condense, to form chromosomes, right? And then metaphase, where the chromosomes are organized right in the middle, where the cell's about to divide, so that the correct chromosomes are going to the respective cell. And then anaphase, where they actually are pulled like a winch from the end of the cells to opposite ends before the cell divides. Telophase is where the DNA unwinds, right, into the respective daughter cells. And then the last stage, cytokinesis, is where the cells pinch off and formalize the process of making 2 separate cells. So that's mitosis. This is a schematic of the IPMAT sequence, interphase. You can see interphase, there's no defined chromosome. There's a nucleolus, but that's beyond this course, but there's just a little bump. Almost like whenever you crack an egg, you can see there's a little bit of a chunk on that yolk. You're always wondering, what is that? Then there's prophase, where the chromosomes start to condense. Remember, the analogy of the rubber band, and you're twisting it and it starts to spiral on itself and becomes tertiary and quaternary spirals, gets thicker and thicker until it becomes dense enough that you can physically see a chromosome on a microscope slide. And then metaphase, now the chromosomes have all lined up on the what we call the equator. And you can see these lines, or they're sometimes called spindle fibers or microtubules. They will grow out and attach to the centromere and then pull the chromosomes to their respective ends in anaphase. And then with telophase, the DNA unwinds. And then, of course, the cytokinesis where things pinch off. This is a diagram. It looks like a photograph. It's what I would call a sketch of a microscope photo. It's really quite nice to look at. The next slide shows you what they actually look like in your lab. So don't expect to see this under your typical onion skin slide. But the question is, can you find the anaphase cells? So which stage is anaphase? So anaphase is where the chromosomes are starting to move to their respective ends. And interestingly, there are 3 photos of anaphase here, and I would say 3 different stages. So we have what I would call early stage, and then we have mid stage, and then we have late stage. So at late stage, the chromosomes have pretty much arrived at their respective ends for the cells. So it's kind of cool. You look at a real onion slide, you can see too, and you should be able to see this under the microscopes in the lab. Here's an early stage anaphase, another early stage anaphase. This is a later stage. I don't see a late stage, but I mean, hey, come on, it's pretty cool. You're looking at chromosomes. So you should be able to see that when you get to your lab. While we're talking about mitosis, I'm going to address another cell division, which is referred to as meiosis, which is a really important step. Now let's break it down. But essentially, meiosis is the formation of gametes. So when plants want to reproduce, they form male components or pollen and female portions, which are egg cells or ovaries that have egg cells. Those are haploid. Because if you think about it, if you have a chromosome from, say, your mom and your dad, okay, so you got 2 chromosomes in your body, and then you want to have a child with someone else, and you have 2 chromosomes and they have 2 chromosomes, and then your child, if you didn't have meiosis, then your child would have 4 chromosomes, and it would just keep doubling, right? And that's not sustainable. You'd end up with so much DNA, you'd just be a big walking test tube of DNA. There'd be nothing much left of you. So in order to reproduce, the DNA has to be cut in half. And we call that, we refer to that as haploid. So normally what happens in a reproductive structure in a plant is that it's identical to meiosis at first, right? So the first half is just like meiosis. You go through the process of cell division, and even here there's telophase, and so you've got 2 cells. But then we reproduce that one more time. Instead of just finishing, we go through a second prophase and we pull the chromosomes apart into halves, right? So just at the bottom, instead of 2 daughter cells, we end up with 4 daughter cells, but each one has only half the amount of DNA. And so each one of those could be a pollen. Each one of these would be a pollen, or they could be an egg cell. So I put together kind of a real simple diagram to help you understand how this would work in a plant. A really good example is begonia. That's the top flower there. They have both male and female flowers. And I've colored the chromosomes, right? So this particular plant has chromosomes from its sort of male and female element, and this female flower has chromosomes from the male and female elements that made it. And they each are what we call diploid because they have 2 copies of chromosomes. Now, they form reproductive structures, pollen, but because it's half, this pollen has this chromosome and this pollen has that chromosome. and let's look at the female. This egg has that chromosome and this egg has that, because you have to cut it in half. And so this might be this parent's gene in that egg and the other parent's gene in that egg, and one parent's gene in that pollen and another parent's gene in that pollen. So we're setting ourselves up for shuffling the genes. And you can see at the bottom, there's 4 different possible combinations of chromosomes when we combine these and fertilize. So 4 potential outcomes. And that's what, the famous monk Mendel basically discovered with peas when he first did his studies on mitosis and the inheritance of genes. I'm not going to go into a ton of detail on this process, but it is important you have an understanding of what meiosis is, what it offers to the plants in terms of their ability to mix and match genes to create new interesting combinations that might be more advantageous to a given environment or not. So mitosis essentially occurs in meristematic regions, or we've just simply referred to them as meristems. Okay, and where they would be located in your typical plant would be in buds and roots and shoots and in wounds, interestingly. If you take a cutting from a plant, the fact that you cut it with a knife triggers meristematic activity. That's why you take a cutting, it will start to callus, and then eventually roots will form. If we didn't have the possibility of a meristem forming from a wound, a plant would never be able to repair a wound, and a cutting would never root. So meristems, it's important for your midterm and your quizzes that you understand that they are in buds, roots, shoots, and wounds. And meiosis, the process of forming gametes, only occurs in reproductive tissues. So ovules or egg cells and in the stamen with pollen. So to review the terms, mitosis produces diploid cells because it has 2 copies, so diploid. And meiosis produces haploid. Think of HA for half, so half-ploid cells for reproduction. Okay, some terms. Um, since I'm talking about diploid and haploid, I want to introduce you to this concept of ploidy, because in horticulture, ploidy actually is important. It's an important term. Um, every living cell has a full set of chromosomes. And I started off the lesson, I talked about how each cell has like a library, and each book in the library can be thought of as a chromosome, right? And each parent contributes a copy of that book. So let's say you've got a Stephen King series of books, and you have the book 1 in the set. You might have book 1 from your dad and book 1 from your mom. So you have a double copy of that book. That's a diploid. And of course, each book contains a series of instructions, right? Maybe how to form a flower, or how to make a leaf, or when to branch. It gets pretty complicated the more you look into it. But there are so many instructions in the chromosomes. But you need all those chromosomes and all the information in all the Stephen King books in the series in order to understand and recreate the plant. Sorry about the Stephen King thing, but but it's important that we have all the chromosomes in order to be viable. So diploid plants have 2 sets of chromosomes, 1 from each parent. Tetraploid has 4 sets of chromosomes, right? That means that 2 sets came from each parent. So you have 4 sets of chromosomes. Remember how I said that if you didn't have meiosis, you would start doubling your chromosome count? So interestingly, unlike animals, plants have this ability to increase their ploidy count, often just through mutations. And in the realm of horticulture and science, we've discovered a way to use a chemical called colchicine to trick plants into forming polyploids. And again, if you went double tetraploid, you'd end up with octoploid. You can see how it goes, 2, 4, 8. It's doubling every time. Now, there's obviously a limit to how many chromosomes are realistic, but certain wheat varieties have, 8 sets of chromosomes. Now, Here's another picture for Derek showing 8 chromosomes in a diploid and 16 chromosomes in a tetraploid. So the X's are obviously chromosomes, and there's 4 sets of chromosomes in the tetraploid and 2 sets of chromosomes in the diploid. But if you look at my little icon on the flowers, it explains why polyploids are important. When you have polyploids, or anything more than a diploid, quite often you have larger and more abundant flowers and fruits. So yeah, when you're growing vegetables and stuff like that, a polyploid watermelon will be bigger than a diploid watermelon. So there's plenty of motivation for breeders to come up with polyploid species. So what are the advantages of polyploid? Larger plants, flowers, and fruit. I already mentioned that. Multiple copies of chromosomes reduce the expression of recessive traits. Let me explain this a little bit. Okay, so in genetics, you have what we call recessive and dominant. And a dominant gene is just like it sounds. It means that if you have a single copy out of all your books, say you've got, 2 Stephen King novels, and one has a dominant gene and the other does not. The gene for that controls for sure what happens. So the dominant gene dictates what will happen in the plant. Let's say you have a dominant gene for a red flower in a gerbera, which is kind of actually is a dominant gene in gerberas. That means that as long as that dominant gene is present, the gerbera will be red. So if you manage to have a situation where the dominant gene is not present, only recessive genes, then you will see the expression of the recessive gene, which might be a yellow gerbera. Okay, so recessive and dominant. Now, sometimes and often recessive traits are not desirable. We want the dominant ones, and we want to make sure that we don't see the recessive one expressed. So if we have a normal diploid plant, there's at least a 1 in 4 chance that the recessive gene will be expressed. And if you're selling seeds, for you to say, look, we have 95% germination, but you should know that 25% of your seeds are going to come out a different, like a smaller fruit, just so you know. Most people are like, well, I don't want that. I want them all to be big. So by having multiple copies of chromosomes, so going to say a tetraploid, you have so many reduced chances of having the recessive gene that you can say, okay, 90% germination and 90% large fruit, because the odds of getting all the recessive genes together in one seed are greatly reduced. Okay, so that's another advantage of polyploid. So the offspring have more than 2 copies of any particular gene, which what we say shields the plant offspring from having deleterious or recessive genes. Lastly, and most interestingly, I think in this lesson is you can make seedless fruit. Here we go. I get to teach you something you might not have known. How do you get seedless fruit? So let me explain. You normally have a diploid plant, right? So 2 copies of chromosomes. And I've talked about how you can have tetraploid plants. I have 4 copies of chromosomes. So a really interesting phenomenon happens if you collect the gametes from one and the gametes from the other, so a diploid gamete and a tetraploid gamete, and you cross them mechanically, just force them to combine. You end up with a triploid offspring because it's got 1 chromosome from parent A and 2 chromosomes from parent B, because you had a diploid and a tetraploid. So 1 plus 2 is 3, which is a really odd number. Get that? It's an odd number. It really is. So if you plant the seeds from a triploid, so it'll grow, it'll flower, it'll form seeds. Okay, because it's got double copies of every chromosome. It's got 3 sets, 4 of each set. That's fine. It's not missing anything, right? So it grows and it flowers, and then things start to go sideways. So what happens? The triploid offspring grow, they self-pollinate, and the seeds abort. Why do they abort? Let's look at it. So what I've done here is I've got the triploid on the top left here. Let me make sure I've got the right thing. So up here, we've got chromosome. I've just pretended there's only 4 chromosomes required for this watermelon to grow. There's more normally, but let's say there's 4. So we got chromosomes 1, 2, 3, 4 from one of the parents, and then chromosomes that's black, and then we got yellow 1, 2, 3, 4 chromosomes from one of either parents, and red chromosomes 1, 2, 3, 4 from one of either parents. Because it's 2 and 1, right? We don't know which is which, but we know we're not missing any chromosomes. So that triploid plant, it germinated, it grew, it flowered, everything's going perfectly. And then when a triploid plant goes through meiosis, we have a bit of an issue because you can't split 3 evenly. So if you look at what I've done here, it's just a few examples of combinations of chromosomes. Remember, you need chromosomes 1, 2, 3, and 4 in order for the seed to be viable. So in the first example, there's no 4. In the second example, there's no 4. In the third example, there's no chromosome 2. In the fourth example, there's no chromosome 4. In the fifth example, there's no chromosome 1. And on and on and on and on it goes. Now, eventually, there is a probability that you will, at some point, randomly get one of every chromosome, which I have, created over here. Which is why if you buy seedless watermelon and grow it, occasionally you'll get the odd seed because there's so many seeds in a watermelon normally that the probability of you getting at least one viable seed is not 0. There is a chance, and so you might get one. And that's how we make seedless fruit. So we cross a diploid and a tetraploid. So try to think of how many seedless fruits are out there, right? And then you think, how do you get the seed from a seedless fruit? Well, now you know. The breeder maintains a parent stock of diploid and a parent stock of tetraploid, and then they plant them in a field, row beside row, and they cross-pollinate each other, and then they collect the seed, and they sell it as seedless fruit. Okay, so let's move on to the next aspect of plant growth, and that is cell elongation. So we've discussed mitosis. I've introduced you to meiosis and the concept of ploidies. Now we're going to talk about how cells expand and grow, and that's using osmosis. I'm sure you've been exposed to osmosis in high school. Again, these videos are in your regular PowerPoint slide. Osmosis, the term here, the movement of water. This is where a lot of times students get confused because they think it's the movement of sugar or whatever. Really, you have to think of it as the movement of water, okay? Not the movement of what we call solutes, which in plants is almost always sugar. So in this scenario, we have a beaker over here, right? And it's got a membrane halfway down the beaker. And there's little holes that are small enough to let water through, but not big enough to let sugar through. And so we have more sugar on this side and less sugar on this side. And the interesting thing about water is that it's determined. It's a determined little molecule. It really is. It's got charges on it. It's got a couple of electrons. It makes it sticky, and it's desperate to get in between the sugar molecules and spread them apart. And so water will move through the membrane over to here where there's more sugar because it's trying to get in between them and spread them apart. And so the water actually lowers on this side as it moves over to this side, trying to get in between the sugar molecules where there's opportunity for their charges to stick. And we call that an osmotic gradient. And what ends up happening is the water will keep flowing, right, until it reaches equilibrium. So if you can imagine, the water level on this side would get down to the point where the sugar molecules are equally spaced apart as they are on both sides of the membrane. And at that point, the water is moving this direction through the membrane equally as it is moving that direction. And we reach what we call equilibrium. So it will no longer have a net change in volume on one side to the other. I'm trying to clean up my little markings. So I have another slide here, an animation that I scammed off the net, which I think is a really cool. In this case, there's no sugar on one side, or they're saying salt, but we'll say there's sugar. and there is on the other side. And so this membrane, you can see the holes. And so water is going to go in and it's going to be attracted to the charges and try to spread those apart, get in between. And in the process, the water volume increases on that side. And the reason we're talking about that is because that increase in volume allows the plant to do physical work. It's like on a hydraulic cylinder. You pump oil into the cylinder and the cylinder expands and it pushes a dump truck up or whatever. And in the case of a plant, it expands the cell and makes it bigger and stretches it out. So the best example I have is in the spring, right? So in this situation with a maple tree, we've got buds that are starting to open. By the way, all winter, those buds are busy. The trees, winter comes in December or whatever, and then, you feel like, oh, nothing's happening. The trees are all dead and nothing's going on. Wait till spring. That's actually kind of wrong. The buds are busy undergoing mitosis and dividing rapidly and forming tiny, 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 densely compact leaves. So the idea of cell division happens all winter. And then in the spring, there happens to be a lot of sugar here, which serves a second purpose. The sugar also acts as an antifreeze so the buds don't freeze. Now, if it gets cold enough, you do get bud damage, right? We've all heard of that, especially with fruits. Like we grow tender fruits in Niagara, peaches. This past winter, there was a lot of peaches that were damaged because it got so cold that the little baby bud inside there was damaged to the point where no fruit would form. But anyways, typically there's a lot of sugar there. And if you think about my osmosis lesson on the previous slide, when spring comes, it rains, lots of water is in the area of the roots and it moves up the vasculature and is attracted to the sugar in the buds. And the water won't give up trying to move into the buds to spread that sugar out until it reaches equilibrium with the sap that's in the tree. And so as water moves in, it inflates the leaves. Like the leaves are literally already divided and they're just inflating. And so this, as you can see, and you've probably seen this before, early spring, the leaves are kind of limp and soft and hanging, and they're being inflated by the flow of sap because of osmosis. And once they're finished inflating and fully formed, then the plant starts to lay down lignin molecules and things like that that help solidify them, and then they become a bit more rigid. So there's some terms with osmosis that you should know in horticulture. Um, you know, the fancy term for a wilted plant is plasmolized. It sounds like something you'd say in like a Star Trek episode. Where's Tom? He's been plasmolized. That the new character for the episode that is gonna end up vanishing or whatever. Anyway, so osmosis is involved not just in growth, but also maintaining water pressure. This is important in plants that don't make wood. So herbaceous plants, if you don't give them enough water, they wilt because they don't have that water pressure to exert on the side walls. And that's what we call turgid. So when a plant is turgid, it's upright and happy. And when it's wilted, it's plasmolized and water's moved out and it wilts. Uh, there is a point of no return, and typically plasmolized is, for a lot of plants, is the point of no return. Flaccid is really your limp plant. When you see a plant that's just drooping its leaves, okay, you can probably recover it. But if you see a plant like this, you've probably done permanent damage or it's dead. And of course, a happy plant is turgid. The other terms, hypertonic, isotonic, hypotonic, those I haven't seen them used too much in horticulture. So how do plants grow? We've discussed mitosis, and now we've elaborated on osmosis, where cells enlarge and elongate for elongation. There are other factors that we might as well introduce in this lesson that affect plant growth. And pruning is a classic example, right? So we've talked about buds and meristems. I've introduced the idea of where meristems are located, like buds, roots, shoots, wounds. Um, Interestingly, if you prune something, we get other branches that will take over as leaders or maybe even just grow. What does that mean? Well, the classic example is looking at, say, a Christmas tree. If you look at a spruce or a balsam, they normally kind of grow helter-skelter like this. They do have a somewhat of a nice appearance, but a lot of times with Christmas trees, they're a nice, perfect triangle because they've been pruned regularly to get that shape. Now, what happens? What's going on here? So it's a process called apical dominance. So think about meristems. I introduced you to the idea of a meristem being a region of active cell division and elongation. The tip of a plant is where we find the apical meristem. And in a dominant apical meristem, it inhibits lateral growth that's anywhere near it. And so you kind of get a classic pyramid shape because branches closer are inhibited, but further away they can grow a little more. The other thing that's interesting about apical dominance, like if you have a situation where the leader is broken, the the neighboring candles will bend upwards and replace it because this apical dominance is broken. They're no longer being suppressed, so that one of them will take off. Sometimes you'll see three of them competing, and eventually one wins out and becomes the dominant leader. So that's apical dominance. So in plants that have apical dominance, we inhibit the growth of other meristems, in this case, lateral meristems. In your labs, you might talk a little bit more about this, but the idea, if you cut the tip off, you break that dominance and your lateral branches will grow more. And so in horticulture, we do a lot of pruning to encourage more branching, to make the plant maybe a bit thicker, to fill it out so it doesn't look so thin. And the hormone that's responsible for that is called auxin. We have a lesson coming up where we discuss plant hormones in more detail, so this is really just an introduction. But auxin is produced at the tip of the plant and it moves down through the vasculature into the plant and has all sorts of effects on that plant growth. As far as apical dominance, there's two kinds. There's complete and there's incomplete. And the classic example is a tree. I mean, you can see it in herbaceous plants too. But in complete dominance, we end up with sort of like your conifer cone-like shape. And incomplete dominance, where there is no one dominant apical meristem, like a big oak, you end up with sort of a dome or lollipop structure where the branches are, each one has sort of its own will, and you have a broader structure. Interestingly, the question is, what would happen if you added auxin to a lateral bud? So if you have a plant and you, instead of cutting the apical meristem, you just buy some auxin hormone and you brush it onto a bud on the side of the plant, that bud will actually expand and stretch out. You can force a branch that might otherwise be suppressed. Here's a picture of a tree that has incomplete dominance. So nice picture. Looks like not your normal commuter cars there.